Hello, my name is Joshua Bacha, and today we will be discussing tumor-associated macrophage subpopulations in intrahepatic cholangiocarcinoma, or ICC. The ICC consists of a dynamic tumor environment where a lot of cells are interacting together to help contribute to tumorigenesis. One such cell type are tumor-associated macrophages, or TAMs. The current understanding is that TAMs are related to M2 tolerogenic macrophages. However, the specific phenotype of TAMs remains elusive. Therefore, more detailed definition of TAMs are needed. Therefore, we aimed to first characterize macrophage subpopulations in ICC using single-cell RNA sequencing. Next, we wanted to corroborate these macrophage subpopulation markers in a validation cohort using bulk RNA-seq. Finally, we wanted to characterize macrophage subpopulations using CyberSort, an immune deconvolution method. All data sets for the single-cell RNA-seq were obtained from the publicly available database GEO. All analysis was done in RStudio using the SURAT package. Finally, cells that express CD14 and CD68 were subsetted and analyzed. Macrophage subpopulations were defined, and a unique gene marker panel for each macrophage subpopulation was calculated. For the bulk RNA-seq validation cohort, it was also obtained from the publicly available database GEO, and differentially expressed genes were calculated. The top 30 genes for each macrophage subpopulation from the single-cell RNA-seq analysis were then filtered if they were present in the bulk RNA-seq differentially expressed gene list. These genes were then graphed as a heat map. Finally, CyberSort was done for immune cell type deconvolution using the R package IOBR, where we were able to deconvolute monocytes and several macrophage subpopulations. The results for the single-cell RNA-seq indicated that there were three main macrophage subpopulations. The tumor-derived macrophages form their own distinct cluster, while the normal liver-derived macrophage clustered into two subpopulations, MS1 and MS2, whereby the MS1, the main genes of theirs, were related to the M1 inflammatory profile, whereas the MS2, the main genes that were expressed, were related to the M2 macrophages. The TAMs differentially expressed their own unique gene markers. Finally, the TAMs and MS1 co-express inflammatory phenotype genes, such as S100A6 and LYZ, whereas the TAM and MS2 also co-express tolerogenic phenotype genes, where it was the C1q and the APOE. Then we did a UMAP plot to show the unique gene markers and the combination, and we see that there's a greater loss of MS2 specific gene markers than MS1 specific markers in the TAM population. For the bulk RNA-seq data, we were able to form the differentially expressed genes and then corroborated with a single cell macrophage subpopulation markers. What we noticed was that an overall trend in upregulation of MS1 pro-inflammatory phenotype gene markers was noticed, whereas the MS2 tolerogenic-like phenotype gene markers was downregulated in the ICC versus adjacent normal tissue. The CyberSort immune deconvolution analysis also showed a decrease. The greatest decrease was in the M2-like macrophages, whereas the M1 had a slight decrease. Furthermore, an M0 subpopulation was significantly increased. In conclusion, our analysis demonstrates that TAMs are able to express both pro-inflammatory and tolerogenic-like genes. However, there is a loss of certain tolerogenic gene expressions that were more related to the MS2 population. Finally, we did identify a set of tolerogenic genes, C1q and APOE, which were persistently upregulated in TAMs and were related to the MS2 population. Studies in other malignancies suggest that C1q production does drive tumor proliferation and is associated with T-cell exhaustion and poor prognosis. The expression of C1q is not that well documented in cholangiocarcinoma and may serve as a prognostic indicator or potential target for immunotherapy. In summary, we were able to show that TAMs in ICC represent a very distinct phenotype, which is different from the traditional M2-like macrophages. Further effort is needed to understand the interplay between tumor-associated macrophages and ICC, with future applications in clinical practice. Thank you.